feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong, in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong, in my happy place. And I'm right where I belong, in my happy place. And I'm right where I belong, in my happy place.
Hello and welcome to the Yonglou Lin School of Medicine's Healthy Longevity webinar from the National University of Singapore. Thanks for joining us, and I want to remind you again to use the Q&A function to send questions. I'm sure there'll be lots of interesting questions tonight, and it'll lead to stimulating discussion. Tonight we have Uri Alon on with us, who'll be giving us a talk about the physics of aging. But before we get to him, we have Guo Zhuyu, who's a PhD student at the NUS Center for Healthy Longevity, who'll be presenting on a published study, Epigenetic Aging and Perceived Psychological Stress in Old Age.
Thanks for the introduction. Hello, everyone. As mentioned, I'll be discussing the relationship between psychological stress and epigenetic aging, focusing on findings from a study published in Translational Psychiatry. Let's begin by briefly understanding what epigenetic aging is. Epigenetic aging refers to how biological age differs from chronological age, which is the number of years that we have lived. This age is computed with DNA methylation clocks quantifying methylation on selected CpGs. Researchers believe these clocks may reflect the impact of external factors on aging. DNA methylation, often referred to an additional carbon group at specific cytosine phosphate guanine sites through an enzyme called methyltransferase. When these sites are methylated, it typically represses gene expression by preventing the transcription machinery from accessing the DNA. However, this turning off is not necessarily permanent. It is reversible depending on various bioconditions and regulatory mechanisms. Psychological stress, particularly long-term stress, is known to affect physical and mental health in older age. It is well known that psychological stress is a significant factor of hypertension, stroke, and heart attack. This brings up the key question of the study. Does perceived stress associate with accelerated aging in older adults? The study involved some 1,000 participants from the Berlin Age Study, which is a large longitudinal cohort study focusing on older adults with an average age of around 75.6 years old. The study used a widely used perceived stress scale to measure stress over the last month. 5 DNA methylation clocks were used to estimate biological age and related acceleration in aging due to stress. These clocks differ in the number and the loci selected for the CpG sites. Overall, both Horvath and Hannum were trained to predict chronological age as first generation, where Fino and Grimm age were trained to predict mortality and health lifespan, known as the second generation clocks. The acceleration is obtained by first regressing epigenetic age on chronological age and taking the model residuals, where positive values often indicate accelerated aging and vice versa. The researchers then fitted a multiple linear regression model to explore whether perceived stress was associated with any DNA methylation age acceleration. For each of the clock, there were 3 models performed. Model 1 reviews a direct relationship between epigenetic age and stress, with Model 2 adjusts for sex, and Model 3 adjusts for additional lifestyle factors. These factors are well-known confounders that may associate with stress and also result in the outcome variation. Interestingly, and in fact, the majority of epigenetic age didn't associate with perceived stress at all. The significant findings were found only in Horvath and female age, but showing an inverse relationship. And after adjusting for lifestyle factors, it indicated no effect. This means that at least in this group of older adults, higher level of perceived stress over the past month didn't translate into faster biological aging as measured by DNA methylation. But even a weak protective effect was identified. This means that the results even indicate that short-term stress could somehow slow the bioaging. The study also found that higher perceived stress was associated with well-known health issues like increased frailty and higher depressive symptoms. Now let's dive into why these results might have occurred. Firstly, the counterintuition may be attributed to what we call biological wear and tear. The effects of aging are caused by progressive damage to cells and body systems over time. Many stress scales, like the one used in the study, only measure short-term stress, while DNA methylation clocks might be more sensitive to long-term or chronic condition. It is also worth considering that if stress is that harmful at all. From other studies and theory, stress at a specific level may even promote survival and decelerates body aging. It also could be that older adults may have developed coping mechanisms that mitigate the effects of stress, preventing these short-term stresses from impacting their biological age. Last but not least, we move on to think of multidimensionality. Stress is multidimensional in terms of duration and types of stressor that induce it. Future study may account for these variations to get a more accurate outcomes. This may include both long-term and short-term stress, also along with the stress at specific time point across one's life. And this is all I have for today. Thanks for staying with me.
Thanks for that. A little bit of a surprising outcome. Maybe we need to get more stress in our lives. Um, we're honored to have with us Professor Uri Alon, the Epigenetic Professor of Systems Biology at the Weizmann Institute of Science and head of the Sagel Center for Longevity Research. Uh, he is renowned for his groundbreaking research and made invaluable contributions to the understanding of systems biology, focusing on the design principles of biological circuits, endocrinology, and aging. His work, which spans both computational biology and traditional experimental techniques, has provided critical insights into network motifs in biologic systems, particularly in the bacteria E. coli, but also in other organisms. His extensive body of highly cited research stands as a testament to the impact he's had on the field. The title of his talk tonight is a physics-based approach to aging, and I think this is going to be a first, Uri, because it's going to be a whiteboard presentation.
Yeah, I like the whiteboard because it slows me down. So hi, everyone, and thanks for the invitation. And the plan, what I'd like to do with you is to talk about how to build a mechanistic model for the damage accumulation in aging and how you can use that model to figure out mechanism of action of mutations in drugs and predict which drugs would compress the sick span or lengthen the health span. And also, conceptually to talk about how you can build models in order to guide new experiments. And the way you build models in physics is that you start with patterns that you need to explain. So let's talk about the nearly universal patterns of aging. So first I'll start with something that's what is not aging, right? This is age T. And imagine you have some organisms living on an island and they don't age. And this is the fraction surviving, but they are eaten up by some predator, let's say, at some constant rate, age 0. That's extrinsic mortality. So of course, the number of surviving organisms would drop exponentially. The survival will be e to the minus h0t, just like radioactive particles. That's not aging because there's nothing different between age 20 and age 70. It's all extrinsic mortality. But if you look at human survival curves or mice or worms, etc., they look different. They look like this. You don't die when you're young generally, and you do die when you're old. So being 20 and being 70 is different. That is aging in our definition. And it's sometimes useful to not look beyond the survival curve, look at the instantaneous rate of death, which is called the risk of death, hazard, the probability to die in a given year. It's the logarithmic derivative of the survival. And the hazard in the case of no aging is just age 0, fixed probability to die at a given time. But if you look at human beings, you get a much more interesting curve. If I do here I want to move this bar here. Yeah. If I plot log age 0 here, so human beings start off dying, let's say 10 to the minus 3 or something like that. Risk of death then drops, reaches a minimum around age, let's say 10 or something, and then it rises, and then there's some deaths by stupidity like this around the teenage, and then there's exponential rise and a slowdown at very old ages. This exponential rise actually continues all the way down if you just look at intrinsic mortality. So this is what's called the Gompertz law, which is an exponential rise in the risk of death. And in human beings, it doubles every 8 years. In other organisms, you see it with different time scales. This rate is more or less constant, and historically in different human populations, the extrinsic mortality is what differs between regions and times. And so we'd like to explain this Gompertz law here. We want to explain the slowdown at very old ages, right? We'd like to explain why it's so universal. How can we calculate those slope from basic parameters? There's other interesting patterns of aging that we'd love to explain. For example, what happens if when you there's research like the kind that's done in Singapore and in other places shows that in model organisms you can have longevity interventions. So you give an intervention and you shift the survival curve so that your T50, your 50%, the age of 50% mortality shifts to higher ages, right? There's something very interesting that happens with a lot of those curves, which is called scaling, which is when you normalize age by the 50%, the median age. So now you survival, you reach 50% survival now at 1, right? Because we normalized out that age. This is 0.5 here. And when I look at the intervention, so suppose this is our intervention, the curves collapse on the same curve. That's called scaling. And it's not for every intervention. For a lot of interventions, like dietary restriction and mTOR, rapamycin, stuff like that, you get scaling. And for physicists, that's very exciting. When you see data collapse like that, it suggests that there's, this intervention is affecting a single time scale and affecting aging at all ages at the same rate. And this was discovered by Nick Strustrup in 2016. And that's the paper that got me interested in aging, because I used to think that aging is very, chaotic and entropic and stuff. And when I saw the paper, I said, maybe it's interesting to do a physics kind of model of aging. So we want to understand scaling. We want to understand the Gompertz law. What else do we want to understand? What happens when you do a longevity intervention in midlife, right? So when you do a longevity intervention in midlife, at least in model organisms like fruit flies, something very interesting happens. This is the age. This is the log hazard, risk of dying. So, these fruit flies, again, they have the Gompertz law. But then you can give them a longevity intervention, which means they die less, right? So this is with the intervention, they die less. What happens if you go from, this is like a better diet. What happens if you go from the bad diet to the good diet, starting at this age? Turns out the sins of the past are forgotten. Very quickly, you go to the better curve. It's not for every intervention. Like if you shift temperature, lower temperature, you also survive more. That just shifts the slope. So some interventions, you get a fast improvement. Other ones, you have memory. Sins of the past are not forgotten. To explain this phenomenon, it shows you that there's 2 time scales, right, for aging. There's a slow time scale for doubling the risk of death, and then there's a quick time scale too. 2 time scales. So we remember that for our models. And finally, we want to explain these shifts. And finally, we know, just because of these survival curves, Look, not all the mice die at the same time. These mice, let's say, are genetically identical and grown in the same conditions, or worms, or flies, or identical twin humans, monozygotic twins. But they die with a coefficient, their times of death have about a 30% variation. And it's not genetic, it's not environmental, apparently, at least not what you can see. What is it? That's called stochastic. There's something about aging that has a strong random component, and that's where physics shines, because we know about random walks and Brownian motion, and we have all this history of talking about stochastic phenomenon and dealing with it. So we want to understand all these phenomena. That's not all, by the way. The incidence of age-related diseases also rises exponentially and drops at very old ages. For hundreds of age-related diseases of different sources. Where does this come from? We want to understand that. If we look at performance like VO2 max or something like that, or renal flow or grip strength, it goes from its optimum, which we'll, let's say, call it 1, to its minimum needed for life, and different things, at least when you do cross-sectional studies, they go down kind of linearly and hit 0 at around age 110. Different kind of things, VO2 max, grip strength. So where does this come from? This is performance, right? So we have 3 definitions of aging: mortality, disease, and performance. In mice, by the way, the curves look different, but also decline. So this is cross-sectional. When you do longitudinal, it looks more like this in individuals. So where does this come from? So we want to understand all this. And I want to tell you that there's been theoretical models since the 50s of aging, and there's a long tradition, Streller-Milman model, and reliability models and diffusion models. And I want to tell you about a model that we constructed, and here's the reference. It's by Omer Karin et al., Nature Communication, 2019. And this is also with some experiments to test it in mice, which can basically explain all of these phenomena and then also teach you new things to do new experiments. And the way we got to that model is by thinking of aging as being caused by accumulating damage, and doing experiments and looking at experimental data and doing experiments where we could measure damage as a function of time in individual organisms. And we used 2 different experiments, one in mice. This is an experiment by the Sharpless lab where the damage, which we'll call X, was basically P16 positive cells, and the mice there emitted light because it was tied to luciferase. These are like senescent cells, you can say, or damaged cells. And each individual mice, you can see the damage rising. And here's another mouse. And this is the damage. And in another experiment, which we did, we looked at starving E. coli. They all die, and we use the dye to see when their membrane has a hole. And so here damage is the membrane potential, you can say, loss of membrane potential. And here, again, individual bacteria show this kind of noisy trajectory, damage just goes up and up. And when they cross a threshold, they die. Okay, so we have these 2 kinds of experiments. And this last one is published last year, the damage experiment by Yifan Yang in Nature Communications, 2023, the coli damage. And both of these, the amazing thing is both of these give the same basic underlying model in the mice and in the bacteria. The model is agnostic to what X is, right? It could be senescent cells in mice, it could be protein aggregates in bacteria. We're agnostic to what the chemical nature of X. What we say is that this damage has production and removal and noise, and then we fit a lot of different kinds of models to see which ones explain the data. And the highest likelihood and simplest model is, we call it saturating removal model. And this is the model. It's a differential equation. I'm going to write down the equation, and then I'm going to give you a kind of metaphor so that everyone here listening can understand what I'm talking about. The rate of change of damage is production, and production in this model rises with age. And then there's also removal, but removal in this model saturates with damage. So damage saturates its own removal. And then there's noise, which is which is white noise, just like Brownian motion. So this is differential equation, stochastic differential equation. When you simulate it, it gets trajectories like this. And I'm going to end, and you die when the damage crosses a threshold. That's a simple way to say death in this model. Now, of course, it's a very, very simple model. Think of this huge simplification that there's a kind of damage. It could be that's fluctuating, and it's in this model driving diseases, driving dysfunction, and that's of course very unrealistic. But maybe as physicists, we always train to look for simplicity. We can now try to disprove it, and if we can't disprove this more and more and more, maybe there is a kind of damage like that. So that's the idea. Now, look, I'm going to give you now a metaphor that really helps me compute with this. So this production term of damage, imagine that there's a village. This village has houses, damage-producing units, and they make garbage. That's the damage. And this village also has garbage trucks that clear the damage, clear the garbage, and everything is fine. I mean, once in a while, the trucks go on strike, which is the noise, and then garbage piles up in the street, but then the strike is over, the garbage is cleared, everything is fine. But this village, houses keep being built, and so there's more and more houses. In fact, the number of houses rises linearly with age. That's this production term, right? And they produce more garbage. And this village is in a weird country. Maybe it's a, I don't know, old style Soviet 5-year plan where you get 100 trucks, but you get no more trucks. Soon enough, it's a huge city, but there's just 100 trucks and they're all overloaded. That's the saturation term. They're overloaded with garbage. And now garbage piles up in the street. And when there's a strike, it goes even more. And it reaches a point where it's incompatible with life. Right? So we have two processes here. A linear rise in the number of houses or garbage-producing units. This you can think of as maybe in this E. coli accumulation of aggregates, or in mice, it could be epigenetically modified stem cells that produce damaged differentiated cells that become senescent or damaged. That's the garbage. And the trucks there would be the immune system that removes the senescent cells. So in each organism, it's a different thing. But the important thing is that you have a linear rise in the damage-producing units and that the trucks become saturated with garbage. Those are the two things you need for this model. And then what happens is each individual has this fluctuating trajectory and can die at different times. When you sum up this first passage time process, you get the Gompertz law. And you even know what this alpha is. It's a ratio between production, threshold, and noise, so you can compute it. And it even shows you the slowdown at very old ages, this process, because at very old ages, production equals removal, and you basically get a noisy process. And so you can explain this entire curve. Of course, not what happens during development, but all the intrinsic mortality and the slowdown. This model also shows scaling. If you do a perturbation that affects production, you basically rescale time and you get scaling. That's very satisfying. But if you change trucks, for example, you don't get scaling. What happens when you change trucks? You live longer. So say I give more trucks, you live longer, but also they die more at the same time. So more trucks, you don't get scaling, you get sharpening. All right, so that's very interesting. Different perturbations, you can tell by the shape of the survival curve whether they affect production or removal. In other words, the shape of the survival curve can tell you what parameter the intervention is affecting. Is it production of damage? Is it removal of damage? Is it noise? Is it threshold? You can tell all of these from the shape. This model provides this rapid shift thing also. Because what happens, let's say, when you do dietary restriction, the houses begin to reduce, reuse, and recycle. And then immediately the garbage is cleared by the trucks. You have less production, and that gives you this fast time scale. The trucks are quick, the rise of the houses is slow, and that gives you all these phenomenon. If you think that diseases occur also when you cross a threshold, you get this incident curve. And the drop at very old ages is because not everybody's susceptible, but at a certain age, everybody has already gotten the disease. And this is a paper where we used Israel's medical records for hundreds of age-related diseases, and also UK Biobank on disease incidence 2021. So you can see how this model explains hundreds of disease incidence curves in cancers, just with the threshold for each disease and a susceptibility for each disease, and the same underlying process. And that's a very, very surprising result, giving us more confidence that there might be a single latent variable, this X, that controls many diseases. Otherwise, it's very hard to explain. By the way, there's an outlier disease that rises much faster than any other, and that's Alzheimer and prefrontal dementia. Both of them rise 20% per year. These rise about 8% per year. Alzheimer rises 20% per year, making us think that there's actually a second X for the brain, potentially. So there could be, like an onion, many layers. The first X is for many diseases, but when you get very old, you expose the neurodegeneration clock, and I can speculate what that is if you like. All right, so where have we gone so far? We looked at the patterns of aging. We looked at a model that's based on direct measurements, longitudinal measurements of damage. Those are very, very important. And E. coli and mice, you come up with the same essential model that has damage-producing units that rise with age linearly, chronologically, trucks that get saturated, and noise, which is trucks going on strike, and a threshold. And that explains all these patterns of aging. Even this one, I can tell you about that if you want to ask a question. And so the performance, and so what can you do with this model, apart from having fun with explaining patterns? As I said, you can use the shape of survival curves to now understand in each organism what is the garbage. Because if you do a mutation and it steepens, you can say, oh, this is a truck. This is a truck. It's not any repair mechanism. It's a repair mechanism that affects that specific moiety that's the garbage. And if I know the truck, I know what the garbage is. So we did that. We started this adventure by looking at an organism where you know the most. And this is thanks to Brian's work on yeast. So replicative aging in yeast, where the yeast, and Brian, you can of course correct me if I make a mistake here, because I want to be I'm a wannabe aging researcher. Yeast buds, and you count the number of generations where you can still bud, and this is generations, and this is fraction that are still budding. And after about, let's say, 20 generations, that's 50% of stopped budding. And you can do an intervention now. Let's say delete a gene, a certain gene, and let's say you live longer, but maybe also change the shape of the curve. And by the shape of the curve, we can know if this deletion, let's say delta PHB1, which has to do with mitochondrial mitophagy. What does it do, right? Is it a truck? Is it or maybe a histone acetylase, or maybe a ribosome? So, and Brian and his colleagues generously gave us their data for thousands of yeast deletions. We're graduate students, if I understand correctly, looked at buds and recorded. The published is the 50% age of lots of genes and double mutations. What they gave us was unpublished data on the shape of the curve. This is the time in which yeast stopped budding. And now we can basically do a genetic screen for steepening mutations or shallowing mutations. Sometimes you get a shallowing mutation. And that genetic screen gives us a biological story that's very clear, you can say. And what it shows is that, in fact, there's two pathways of dying, two kinds of garbage. One of them is histone acetylations that make subtelomeric regions and other closed chromatin open up, causing aberrations such as R loops, which is bad hybrids of RNA and DNA. And that causes double-stranded breaks by replication fork collisions. So that's one pathway of dying. And we know from Nan Howe's work that yeast actually can die in two different ways, with round buds or with elongated buds. And consistent with that, we found another pathway buried inside these genes. One is that protein misfolding gives rise to misfolded proteins that create aggregates. And these are removed by many different kinds of trucks, but the trucks that get dammed up, apparently, are for removing aggregates in the mitochondria. It's like having a garbage disposal that gets gummed up first, whereas your big garbage can doesn't. So those are the trucks. And that causes all kinds of problems. Mitochondrial damage can cause all kinds of problems. So these are two ways of dying. And if you get rid of both of them, there's a third way of dying that we have only clues from, like very something that has to do with metabolism, amino acid metabolism, we don't understand. And when we think about human beings or mice, we think that some of these genes translate to progeria genes. For instance, the R-loops genes are some of the trucks are homologs of Werner and Bloom, which are progeria genes in humans, and also lamin A, which in humans it was progeria and controls this chromatin. And double-stranded breaks, we know in lots of long-lived mammals have better double-stranded break repair, et cetera. And in neurodegenerative diseases, we have this pathway. That's maybe the second clock. So we want to think that this yeast stuff tells us about the cellular modes of failure, but humans and mice, of course, also have an immune system and an endocrine system, etc. So we think that these two pathways lead to damaged cells and senescent cells, that cause inflammation and stem cells exhaustion. And we think that in mice, this is the X. And what's the trucks? It's the cells that get rid of those damaged cells, primarily NK cells and macrophages. Those are the trucks. And of course, you don't have an infinite amount of NK cells. They don't grow with age so much. So they get overloaded. When you're young, those damaged cells are good for you. They're good for regeneration. You have enough trucks. Trucks are there from evolution. We didn't evolve to be old, so you don't get more trucks. Maybe you can't even when you're old. And now when you're old, you have so much damaged cells. Your NK cells are overloaded. You have a lot of inflammation. If you get cancer, those NK cells can't reach it in time because they're overloaded. Risk of cancer rises exponentially with age. If you have an infection, those NK cells can't kill the virus infection cells. Risk of dying from COVID, that rises exponentially with age. So all these factors get into this in this paper, explaining how you can get these threshold crossing events that lead to diseases with age. So that's one use. Shape of survival curve tells you what parameters you can, for, let's say, deletions, interventions, kind of deduce what part of the mechanism they affect. It also tells you how to combine interventions, because you want an intervention that affects production, combined with an intervention that affects removal, and then you get the best of both worlds. You get scaling, long life, and you get steepness, which means a more equitable age of death. We can also tell from this model that if you think about health span, if you want to shrink health span, I know I'm telling you a lot of things, so we're almost at the end. If we want to expand health span, that's to say, compress the fraction of time you are disabled divided by your lifetime. If you want to do that, from our model, since we know about diseases and death, we can predict that steepening interventions compress morbidity. And scaling interventions, so you can tell from the survival curve, they're going to scale morbidity. So you live longer, let's say by 10%, but you're also sick longer by 10%. But if you combine a scaling intervention like rapamycin, let's say, with a steepening intervention like a carbose, you get the best of both worlds, longer life and compressed morbidity, according to this picture. And we know from invertebrates that indeed steepening interventions compress morbidity in flies, worms, mice. And that paper, I just want to mention, in case you'd like to look it up, is by Ifan Yang in Nature Communication in revisions, and you can look it up in bioRxiv in 24, about compressing. So this can tell you which interventions to combine and which interventions maybe to prioritize, just looking at their survival curve, because they're more or less likely to compress morbidity. So these are some uses of a model. And of course, in my opinion, a good model is one that makes you do a new experiment. And we're having a lot of fun now with this model, thinking about heritability of aging and thinking about testing these mechanisms experimentally in humans. And I'd love to hear your questions and to continue to engage with physics of aging. I want to invite physicists, computer scientists, engineers to go into this field. We need you. What you know can help us. And it's a very exciting and very satisfying field to be in, because it's just exploding now with new technologies and understanding. So thank you.
Thanks a lot. First of all, I want to remind everybody, put their questions in the Q&A and we'll get to those. Um, well, first, let me reiterate what you just said, which is that we need more people like this in the field. The longevity field has really exploded over the last couple, maybe the last decade. A lot more clinicians, there are investors, there's a lot of other computational people, but we need physicists and other theoretical people as well, among other groups. And actually, we're going to sponsor a meeting on the physics of aging in Singapore sometime early next year, which you have to come to. So don't even think you have a choice with that.
I'd love to. And the work you're doing to bring together theorists is superb, because being together is much better than working alone. So thank you for that.
Well, and I think this next discussion is going to reflect the fact that I need to be around more theorists. So, um, coming back to the yeast, one of the things that struck us, because we were screening for deletions that live longer to start with, um, is we ran into this barrier. We could make one mutation and they made 25% longer, and then combine it with another mutation and we get to 50% longer. And then at that point, we couldn't combine anything and get past that. And my thinking on that was always that, there are limitations on survival that come into play at 50 generations, but not 25 generations. And the mutations you make at 25 generations are not going to uncover that 50 generation pathway because they're not relevant at 25 generations. And I think that's what you're starting to get at. You find two mechanisms in yeast, and then maybe a third one becomes relevant. And if you could fix that, you would get to a fourth one probably. So I suspect that mammalian aging is going to be quite similar to that. That if you break down some barriers, you're going to encounter new barriers. They may be more interrelated. I don't know, and the immune system definitely may connect them together in in more ways. I wonder what you think about that concept.
I agree with you. I think one saving grace of aging research is that, unlike the time of life where evolution is working very hard, which is development and growth, there almost all processes are equally important. Because if one is, less good, evolution would work to make it better. And so everything is important. When evolution is no longer acting very strongly like in age, it's possible to have a dominant and a subdominant and a third. There's no force pushing them to be equal, you can say. And that's why I think there's a chance that we'll uncover like the first layer in the onion. So in yeast, the first cause of death is this mitochondrial one. Once you solve it by, let's say, HAP4 deletion, by some transcription factor, the yeast actually make a toggle switch decision early on, which repair pathway to turn on, and they die from the other thing. So when you turn on that repair pathway constitutively, you die from the second thing, which is the histone acetylation path. And when you cure both of them, you die from a third thing, which we don't understand. So I think it'll be a layer like that. In human beings, we already know that if we live long enough if we survive cancer and heart disease, we'll get into neurodegeneration, right? And after that, we'll get into the next thing. So I think we should think about it like that, onions. Another thing about the maximum lifespan, in yeast, you can expand maximum lifespan, like you said, by 50% and more. In humans, it's not clear that it'll be the same, because historically we haven't, so a lot of things kind of slow down around 100, and there's never been a human being probably lived up over 120, even though they're meditating on a mountain and eating vegetarian food, and okay. So it could be, this is my thinking, very naive, that humans might be in the clade of primates optimized for slowest development and therefore longest life. So we're already optimized there. Whereas things like other monkeys aren't. In the field of worms, you have C. elegans that's not optimized, but you have gut intestinal nematodes that can live for years. So in its clade, you can push it. So maybe in each clade, there's something that's like your chassis can stand a maximum lifespan. And in order to extend human maximum lifespan, we'll need to do a deeper redesign. But shrinking morbidity is much more within reach, in my opinion. So this is very interesting points for why are mice so plastic with their maximum lifespan, yeast, but humans, it's not clear yet.
Yeah, I think that's true. And I've been saying for a while now that I think that the interventions we've discovered in humans, or I think a lot of them will work. Well, we discovered them in mostly in animal models, and we're trying to apply them to humans. I think that a lot of them will work, but they're going to square the curve and maybe push out health span
Yeah.
some. But whether they affect maximum, I don't know. And so, Peter Fedichev and I talk about this all the time. We think that it may be that we have to think completely differently if we want to have huge effects on maximum lifespan.
Yeah, and that'll be an amazing conversation to have in your meeting.
Yeah, I think that, and he's thinking about what kinds of interventions might do that. Does it have to be damage? I mean, you modeled everything on damage, but there are other theories for things that can drive aging. I'm thinking of like Mikhail Blavatsky, who unfortunately died recently.
No, really? Oh, I'm sorry to hear.
Yeah, great thinker in the field.
Yeah.
It's going to be missed for sure. And his theory was like hypertrophy, which pathways that were important for signaling during development get hyperactivated or not turned off during adulthood, and they continue to, try to drive things like cell growth and proliferation when they shouldn't be going at that level. And that's would be another explanation for what mTOR might be doing, and that might be driving aging. So I'm wondering if you've presented a really elegant model that's relying on damage, and of course damage can be defined a lot of different ways too. But does it have to be damage that's driving that?
No, it doesn't. So this is like they always say theory is a fiction. It's a useful fiction. Useful because it makes you do no experiments. And the more you can try to disprove a model, and you can't, the more confidence you have, you can never prove it. So we use damage because that seems to be, supported by many experiments that if you increase damage, certain kinds of damage, not all kinds of damage, but certain kinds of damage, you have accelerated aging, and if you slow it down, you can have slowed down aging. So it might be causal, but if you think about Blavatsky's concept of hyperfunction, let's say mTOR is working beyond its normal design. The question is always why? Why does that happen at age 70, not 20? So you need some ultimate cause. And it could be, for example, let's imagine you have, if it's a multicellular organism like a mammal, maybe you have these pathways that describe making damaged cells, making inflammation. Now you have rising chronic inflammation. And aging, in certain respects, phenocopies responses to inflammation, stem cell exhaustion, turning on those pathways. So the cells are good citizens. They're acting like they should. They're thinking there's inflammation. They're thinking there's loss. They're acting like in the young, like they should. But when it's chronic, and that you begin. So I think that Blavatsky's concept integrates with damage theories. They're not exclusive, and I think it's very useful because I think philosophically, there's also a possibility, of course, that aging is programmed, as that we evolve to age, get rid of old professors to make office space for young faculty, which, it of course might happen in certain restricted situations, but in simulations it doesn't appear robust enough to explain aging. But of course, Blavatsky is saying evolution has given us this highly designed response systems. And what happens in aging is they're working, but just outside of their design range. Too strong, too long, for too long, wrong places. And then we need to ask why. It's not just like a car that breaks down. And that's how you can integrate damage. But of course, we have to really be agnostic that we don't know the answer yet, and work with parallel models and compare them. Can they address all these quantitative patterns? Can they make us do a new experiment on, let's say, longitudinal damage measurements in mice or whatever, in humans? Challenge ourselves, right? So there used to be a Ross theory of aging, but people actually at Singapore, like Jan said, but Ross doesn't rise exponentially with age, so it can't be, right? So you can actually discount certain kinds of damage, and it's all testable. That's the important thing. Did I answer your question?
Yeah, I think combining the theory with the testing is what's so profound. And maybe I'll ask an annoying question.
Yeah.
Is this the only model that really fits the data, or are there other kinds of mathematical models that equally well fit the various observations that, I mean, how unique is this model?
So it's not unique. You can add things to it and still get all the qualities. It's just you add more parameters. For example, if X generates more X, so the production is not just eta t rises with time, but eta t plus A plus X. So X makes more X, like maybe senescent cells have a bystander effect. You still get all these properties. It doesn't ruin the model, but you can't. Okay. Another possibility is trucks I portrayed as just being constant with age. But what happens if trucks get rusty, degrade with age? Like maybe the immune system, we know certain aspects of the immune system, adaptive immune system particularly, gets degraded with age. That also, if you add that to the model, you also can explain all these patterns. It's just an extra parameter. So we can't, what we like to say is the simplest model is what I showed. And then damage can produce you can rule out, for instance, that House's eta rises t, but if they would have rises t squared, the Gompertz law would not be e to the alpha t, but alpha t squared. So we can rule out a lot of stuff. We can't rule out additional complications, you can say, or additional phenomena. And then there's other models that are like reliability models. They're just completely different assumptions. Like we have multiple organ systems, and when one fails, it makes the other ones fail more. And they can explain some of the patterns, but not all of the patterns I showed. So you can now start comparing models. And of course, if you add more and more parameters, you can explain patterns. And then it becomes an experimental question, right?
Yeah.
And so one way of methodologically working that's a little bit opposite of biological training, where we try to find complexity. Like if we found ERK1, well, look, here's ERK2. Oh, 3, ERK4. Here it's to say, oh, there is ERK1, ERK2, ERK3, ERK4, but let's find the model that's the simplest model, but no simpler, that can explain the essence.
Yeah.
And then if we need to ask another question, we can make it more and more complex. So it's like, I think we need to approach this with a hierarchy of models where this is the simplest, most understandable, and then you add more and more and more reality, truth, facts, until you get to a full description of the human organism with hundreds of equations. We'll have no chance to understand it unless we know how to break it down into modules that we do understand. And so that's a kind of approach. I hope I answered your question.
Yeah, no, I think it's great. Maybe I'll ask one more and then we'll see if there's questions. I'm taking more time tonight. People think that diseases drive aging, and certainly that certain diseases are associated with earlier mortality, even through other mechanisms. And so does that fit into the model somewhere? So when you get a specific disease, say diabetes, does that accelerate aging in the model, or can you fit that in easily?
So I think at the level of simplicity that we're here now, there are diseases and mortality are independent. So the way we model diseases, but of course this is not realistic. It's just a modeling choice is that you have this latent variable, this biological age, this X, that's rising and dropping stochastically quite rapidly and rising over the decades. And that's our holy grail. We want to know what X is for a given human being. And when it crosses a threshold, if you're susceptible, you get a disease. For example, the inflammation causes insulin resistance. Not everyone's going to get prediabetes, but if you have low beta cell carrying capacity, and you're eating a lot of sugar, that person has a threshold, and prediabetes is going to occur at that X level. And then, so in a more realistic model now, because glucose is high, that can impact the model parameters, and the person is going to get other diseases more quickly, et cetera, et cetera. So we are interested very much. This is why I said there's a huge range for theorists here and for computational scientists. Want to get at the end to a personalized health model for a person, given your history, like an LLM, find out what's going to happen next. So if we know that diabetes has a low threshold and you hadn't gotten it yet, you're probably not susceptible.
Mm-hmm.
And if it's linked to another disease of old age and you haven't gotten it early, you're not going to get that disease of old age with high probability. So from this kind of way of thinking, you'll be able to have predictive models and prevention models. So we think the key is knowing that latent variable, and also that'll be the ultimate biological clock once we know how to measure it. What do we need to measure? Histone acetylations? Is it mitochondrial aggregates? Is it senescence? What is it that's likely to be common to all aging people and radiating out according to your genetics cross environment to see if you have a threshold for different diseases? That's a simplifying picture. Of course, the reality is much more complicated, that we hope to exploit to make as much headway into personalized aging trajectories.
So I'm going to bring Tim Vanas in, who's here to handle questions. We have less questions tonight. I think equations have scared people, but it's more time for me, so that's fine. Tim, how are we doing?
Very good. Thanks first of all for the great presentation. I found it really interesting.
Thank you.
There are no questions as of now, I think, but I actually have one of my own. So initially you showed these three graphs with mortality, disease, and then also performance. And I think you also mentioned that the performance curve was linear. I was wondering whether the model that you created in that sense is also valid for performance.
Yeah,
that would be a different model, or
I'll tell you some unpublished stuff. So when you do cross-sectional studies on grip strength, VO2 max, you get this kind of curves, where 1 means typical performance at age 20, and 0 means minimal performance compatible with life. And you get that data collapse. Everything in the model doesn't look linear. Damage looks like this. Because at some point, you have more houses than trucks, all bets are off. So how do you connect these? So it turns out that if you assume that performance drops with damage, just like Michaelis-Menten, and you average that, you do a cross-sectional study, you get this. Because, just mathematically, that function goes like t, something like that, okay? That function is very similar to this function. It's 1 minus, okay? So the math here, when you average, you get it automatically from the model. And we're very excited about that, because inside this nonlinearity, if you have performance that drops with damage, but evolution designed it so that K is the amount of damage you reach in the reproductive years. That's the halfway point of removal. If evolution designed it so that when you're young, the damage doesn't affect performance that much. But when you're old, X is beyond what it should be. Then damage, so you get it. And if you look at individual longitudinal trajectory, you get this kind of things, which are what you measure longitudinal. So we're very excited at the model. So this is another thing. When we see a pattern like that linear thing, we get very excited and very scared, because we say, oh, we might have killed our favorite model. And then in this case, we say, okay, and then we're more excited. Doesn't prove the model, but that's the way we work. So we try to always test it. Did I answer your question?
Yes, you did. Thanks.
Bruce Zhang says, thank you for this great talk. How do such physiological models explain changes in inter-individual variation in lifespan? So what Bruce is asking is, how do these models explain the fact that genetically identical organisms have different lifespans, if I got it right? Do you agree that's the question? Inter-individual?
So, yeah.
So the way the model explains it is that every time you simulate this equation, because of the noise, you get a different trajectory. And then you can ask, when does it cross the death threshold? And you get the different individuals cross it at different times. And those different times have exactly the distribution that you observe. The Gompertz law. So what does it mean? It means that this fact that trucks go on strike. And the way we interpret it in mammals, by the way, is the immune system goes on strike. What does that mean? Bad sleep, chronic stress, poor nutrition, infection, injury. All these things make the immune system focus on other things and removing senescent cells and then go back. So we think that kind of thing might be a candidate for the noise. And that means that really jitters the trajectories. That's just a hypothesis, right? We don't know what the noise is actually. We don't know what this epsilon is. We'd love to know. And that stochastic the houses and the trucks are very deterministic. But if the trucks go on strike, garbage piles up in the street. Now they go back. It goes back on a fast time. So this kind of chaotic process, we think, drives the stochasticity in diseases and in mortality as a kind of a conceptual picture. Bruce, I hope I answered your question. And can I keep reading, or do you want to read it?
Yeah, I can go on with the
Go
second
ahead.
one. So
go ahead.
Steve-O asks, what are the biggest influence of aging in this model? Also, can you send us a link to a lecture with better notes? And I think
Yeah.
it was referring to factors such as inflammation.
Yeah. So I want to first give you a link. First of all, I want to give a plug for my recent book called Systems Medicine. This is a periodic table of diseases. And the last 3 chapters are basically this lecture. So you can see it all there. Also, if you don't want to buy the book, there's basically the same thing free online, which is my Systems Medicine course at Uri Alon website. And it's video plus text, all annotated like this. And then you can see the same approach for endocrine systems, diabetes, fibrosis, autoimmune diseases, and aging on this course. So you can see in detail, and I also do some guitar songs there. It's a lot of fun teaching kind of thing.
Yeah, I think you missed your career. You should have been a guitar player.
Yeah, you say, what you're telling me is quit your J job, right?
Never would I say that.
Yeah,
Tim, I think
thanks
there's
though.
one more question. Yeah.
So
Meg
biggest
Morris.
go ahead. Inflammation.
Meg
Oh,
Morris asks, thanks for the great talk. To what extent does exercise slow the rate of damage?
Meg Morris. So here I think Brian is more equipped to answer. I would just say maybe Brian can contribute.
Yeah, I mean, I think a lot of these lifestyle interventions, they're both either slowing the rate of damage or improving the repair of damage. So they could be affecting the production of the garbage or the trucks picking up the garbage. And maybe both. And so I think that modeling how they fit in would be interesting, but I think that probably they're multifactorial.
And I agree. And so, but we have now a way to tell. So if you look at survival curves of exercising mice or exercising humans, if they shift in a way that scales, we say, oh, they affect production. If they shift in a way that steepens, we say they affect trucks. And you can tell if it's both, and you can tell if it's noise. So we have a way into this question. By the way, I don't know the answer, and this is very fascinating. Meg, I'm going to run and do it because I think there is data like that in mice at least. And exercise is one of those beautiful things that affects many different processes, all beneficially, I would say, unless you over-exercise. It lowers inflammation, insulin resistance, and it increases vasculature. It increases blood volume by 10%, which means the trucks have more roads to get there. It helps cognition. So it does something also probably to the garbage trucks in the brain, which we think are glia that can eat up damage from the neurons and reduce neuroinflammation. And astrocytes can transfer fresh mitochondria into neurons. All these wild things happening in the brain. So exercise apparently has a big effect. The mitochondria signal to the brain with all kind of hormones, and it's. I think it's very important to study how exercise does that, and maybe we can have a clue with our survival curve.
Well, we're running a little bit late, so I think I'm going to have to cut the questions off because we're over an hour already, and your fee's too high, Uri. We can't keep you any longer. Yeah, I want to also add that usually if I say a model is garbage, I mean a bad thing, but in this case, your model is garbage and it's brilliant. So thanks a lot for sharing it with us. We'll look forward to seeing you next year in Singapore.
Yeah, I look forward also. Thank you.
So Thank you. I want to remind everyone to use the chat function and panelists and all attendees option to leave comments and feedback. Our next webinar will be next week. We'll have Sebastian Brunauer, who's the CEO and founder of ImmuneAge Bio, and his topic will be building and investing in longevity biotech companies. I want to leave you with a final video of Choi Soon Hwa, who's the 80-year-old Miss Universe contestant. Thanks for joining us, and I'll see you next week.
그러다가 제가 미스 유니버스 나이 제한 없다는 걸 보고 나 여기 나간다 그랬더니 어머니 거기까지요? 어머니 자관 뭐 하고 싶은 대로 해보세요 그랬거든요. 그래서 제가 이제 어저께 사진도 붙이고 했는데 대단합니다. 우리 어머니 대단합니다. 우리 할머니 짱 그랬죠. 그래서 제가 굉장히 기분이 좋아요.
Korea is having a very fashionable moment with the old age. You'll see it in the advertisements, on the TV shows and fashion shows, and even in the international beauty competitions.
가지고 있던 재능 그런 것을 자기 자신이 키우기 위해서 사회에 나와서 자기가 가고 싶은 어떤 그런 곳을 다니면서 굉장히 그 활발한 활동도 하고 또 그렇게 함으로 해서 우리 시니어 사회가 굉장히 활발해지고
I'm feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down.